you've seen this. I want you to know how to contact me, drg@readysorb.com, and the phone number below for any questions, and we can supply you with references and more information. Several studies have reported that exposure to EMF results in increased oxidative stress in the tissues of the body. To me, this equilibrates to diminishing the reduced glutathione, the amount of reduced glutathione available for body functions. In honeybees, this translates to increases in their superoxide dismutase, which is necessary to take care of the superoxide radical, and the results of splitting oxygen into superoxide and hydrogen peroxide. So glutathione is manufactured in the cell of the body, although it's needed in many areas in the cell. It's essential for the function of the mitochondria. And to facilitate this, there are two pumps that are genetically expressed in the mitochondria to carry GSH, the reduced form of glutathione, into the mitochondria to help reduce the excess free radicals formed in the electron transport chain. That's the ETC. So glutathione is resistant to most enzymes in the body, but glutamyl transpeptidase found inside the cell is the only one that can break glutathione down. The GGT is actually formed, then expressed on the surface of the cell, and it's used to break circulating glutathione down into its constituent parts, and we'll talk about that. Glutathione also works with transferases, which are used for detoxification, peroxidases, which are used in eliminating hydrogen peroxide and the OH radical, and the reductase enzyme, which can transform oxidized glutathione back into the reduced form. Because there's not enough of this reaction occurring, new glutathione needs to be formed on a regular basis, and this is done using two important enzymes, glutamine cysteine ligase and glutathione synthase, GS. The problems occur when the GCL is not expressed adequately. It actually requires two modifier units, a catalytic unit, a modifier unit, in order to adequately express GCL. And there's certain conditions in which GCL is deficient. HIV is well known to prevent this expression, but it also turns out that type 2 diabetes prevents the expression of GCL, and so these folks have diminished glutathione. We're coming out with a paper that shows we can restore glutathione to functional levels in people with type 2 diabetes, but this has been previously examined in a preliminary paper, which whose PMID is listed to the right there. Lupus erythematosus surprisingly has a deficiency in the expression of GCL, and I've not seen any papers that explain why that is happening, but that leaves these people with a type 2 immune response, which is accompanied by the formation of autoantibodies. Also, those of you interested in mold and mold toxins, mycotoxin exposure can lead to diminished formation of GCL due to lack of expression of the GCLC catalytic unit. And this on the bottom right is a reference to the paper that Dr. Jeanette Hope and I wrote several years ago describing this phenomenon. And I think that's an important component of mycotoxin illness. Let me know if you want to set up a small webinar with a few of you on mycotoxin illnesses. Moving forward, this paper described the fact that liposomal glutathione will work in people with HIV, and decreased formation of glutathione will work, an amount that is needed to form adequate glutathione is 1,000 times lower than the amount of NAC that is required to accomplish the same result. So another way to say this is that liposomal glutathione works 1,000 times more efficiently, but the scientists like to say that the LGSH, liposomal glutathione, at 1,000 times lower amount will provide the same result as NAC. So here we have a picture of a liposome with active glutathione in the center. And we talked about the fact that it's able to replenish glutathione very efficiently inside macrophages, for example. And they've done this both directly in cell cultures and demonstrated that liposomal glutathione will be readily absorbed and restore macrophage glutathione in humans in 2 separate studies. In adults, and here's the studies. Dr. Lai and company did the first paper, and a second paper repeated the study and showed that T cells in people with HIV have lower levels of glutathione, and that up to a point this can be restored, but when they have extremely low CD4 T cell counts, it's hard to restore the glutathione there. Dr. Janet Kern several years ago did a study in children with autism who also have deficient glutathione, and demonstrated that in children you can safely and efficiently restore serum glutathione back to normal using liposomal glutathione. The study that I mentioned by Lee and Valdivia, this is a diagram that shows that during this viral infection, glutathione is diminished and you get an excessive production of IL-6. This may come into more focus with the next virus we can talk about. Anyway, if you can, with the oxidation stress, you get a decrease in a variety of cytokines, the immune hormones, and an increase in TGF-beta, as well as an increase in IL-6, and a decrease in several of the cytokines that are needed for microbial activity in macrophages and things like that. So then, after the ingestion of liposomal glutathione, IL-6 was decreased and TGF-beta was decreased, both of these going back toward normal. The immune function was also improved dramatically, and when they took organisms from Mycobacterium tuberculosis and infected the macrophage cells from these individuals, they were not able to replicate efficiently at all. There was almost no replication. So that's the conclusion of the information that I had planned. Oh, no, we've got 2 studies with another virus that is topical right now by Guilford and et al., in which I was able to participate. That demonstrates that glutathione becomes deficient in individuals exposed to COVID. This has been published in a small study from Russia by a man named Politnikov, and I would urge you to take a look at this study. And there's an addendum, and the combination of those 2 shows that both IL-6 and TGF-beta are significantly increased, and separate clinical studies in each of those materials shows that they are significant markers for severe COVID disease in humans. And so these are referenced in both the primary study and the addendum. The primary study can be found by plugging in the words Guilford, glutathione, and COVID-19. And the addendum can be found by plugging in Guilford addendum and then the numbers 6357 into a search engine. If it doesn't come right up, you could add the word encyclopedia, because they're publishing this addendum in an encyclopedia. It may come up just with encyclopedia 6357. So I appreciate your attention. If you have any questions, please let me know, and we can talk more about this at your convenience. Thank you.